For those of you who don't know me, hi everyone, my name is Kai. I'm working at the Cecile and Oskar Vogt Institute at the University of Düsseldorf in Katrina Amunds lab. And I'm very delighted that I have the opportunity today to give you a talk about high-resolution cytoarchitectonic maps of the medial geniculate body in the big brain and how we advanced to create these high-resolution maps. And as you can see, it's more about brain mapping, so more on the neuroanatomical side. And to give you a quick introduction of how we usually perform brain mapping, we usually cut brains into sections. You are all aware of that. And when we've done that, we need a method to actually quantify the cytoarchitecture that we can see after it has been stained for cytoarchitecture. So the current gold standard method for doing so is the Schleicher method, which extracts, which has been developed in our institute more than 20 years ago. And what the Schleicher method actually does is it extracts profiles from the cortex to be able to detect borders between cytoarchitectonic areas. And it has some major advantages. So it's very reproducible. It's also relatively robust. And it does the border detection based on image analysis and statistical criteria of profile shapes in comparison to a mere subjective way of visual inspection. So this is a very big advantage, but it also has some disadvantages. First of all, its throughput is limited. It takes roughly 1 year per area per brain on every section of a data set to map a whole cortical area. It's also not applicable for 3D data. And most specifically important for this talk, it is also not applicable for subcortical nuclei. So you can already see that depending on what you're trying to map, the current gold standard method is often not feasible for the big brain dataset. And this is why Christian Schiffer, whose talk you've just heard at the beginning of the last session, has developed a deep learning-based tool to overcome some of these limitations. And he's already shown you that it works quite well and is able to create high-resolution maps in the big brain. What we were then interested in after we've seen these first great results is how does this deep learning-based tool actually operate? And what we've been able to find is that there are actually resemblances inside the tool's architecture that resemble well-known cytoarchitectonic criteria that are used by neuroanatomists to denote differences between cortical areas. The tool and the architecture, I should say, of the tool learns to resemble cells, it learns to resemble layers, and it also learns to resemble cortical areas. So we think that the tool is actually quite a valid semi-automatic alternative to for the current gold standard and helps us to map in the big brain when we want to create high-resolution whole brain maps. So to sum it up to now, we now have a deep learning-based tool that operates on cytoarchitectonic criteria and helps us to create these whole brain maps of the cortex. We can then in return use these maps and link it with functional aspects of brain organization. And this is especially important to note because this is what big brain allows us. Because in comparison to other atlases, the big brain provides a unique histological reference frame that allows us to do this comparison. And if you haven't had the chance yesterday to join the hands-on session of the BigBrain Warp Toolbox by Casey Pocola, I would just very much encourage you to do so and to take a look at the video if you want to get a more in-depth look on this, how you can combine the microstructure of the big brain and functional aspects of the brain organization. What we then were interested in the next step for us was to see whether we could also use the deep learning-based tool to create maps of subcortical structures in the big brain. This is becoming increasingly more important as functional measurements of, for example, ultra-high field fMRI have broken the sub-millimeter barrier in the last years. So now functional linkage with subcortical nuclei becomes feasible. And this opens up a whole new space and new possibilities for microstructurally informed brain models and simulations, for example, that can integrate and rely on the big brain data. And this is why we've started a new mapping project in the big brain that wants to shed some light onto this. And this is why we've started to create high resolution 3D maps of the human metathalamus. The metathalamus is actually comprised of two nuclei, the lateral geniculate body and the medial geniculate body. And the reason why they are so important is that they constitute, and the reason why we've chosen the lateral and medial geniculate body for this approach is that they are important subcortical relay stations for visual and auditory information. On the left side, on the right image on the bottom left, you can actually see the 6-layered structure of the lateral geniculate nucleus that was mapped by Andrija. And on the right side, you can actually see the three subdivisions that I have identified in the big brain that belong to the medial geniculate body. Medial geniculate body. And the lateral geniculate body is a relay station for visual information, and the medial geniculate body is a relay station for the auditory information. And what we've done, basically, and this describes the workflow is, we've created delineations of the medial geniculate body and also the lateral geniculate body on a subset of sections in the big brain based on cytoarchitectonic criteria. So I've actually mapped the medial geniculate body and its 3 subdivisions on roughly every 5th section of the big brain data set. And then in a second step, we use these delineations to train the deep learning-based tool that Christian Schiffer has introduced you to at the beginning of his talk, and then use the output, the predictions of the deep learning-based tool to do a 3D reconstruction and create whole brain cortical maps of the medial geniculate nucleus, and in Andrija Brandstätter's case, also the lateral geniculate nucleus. And what we, in the end, came up with is a very detailed view of both nuclei in the big brain. As you can see, we're zooming into the big brain right now and zooming into the thalamus, more specifically the metathalamus, and these 2 structures pop up. These are the 3D reconstructions that we were able to create. These are whole brain maps of the medial and lateral geniculate body. On the left side, you can see the lateral geniculate body and its 6-layered structure. And if you look at the histology beneath it, you can actually see the different layers of the structure. And as it is blended in right now, again, on the left side, on the right side, you see the medial geniculate body, and it's fading out in a second now, and also reveals the underlying cytoarchitecture. And we hope that with this work, it will become possible to link some aspects, some functional aspects that these 2 areas and these two structures are involved with the underlying cytoarchitecture. We also hope that we might be able to inform modeling and simulation. And and we look forward to actually apply this also to other subcortical nuclei that are of interest. And with this, I would like to end my talk. Thank you for your attention. Thank you also to my colleagues. Thank you to Katrin and Andrea and Christian specifically, but also to Sebastian, Hartmut, Mona, and Philippe, who also participated in making this possible. And yeah, I see that I stayed under the 15 minutes, so we might have some questions that I hopefully will be able to ask. Otherwise, you can also contact me, drop me a mail, or yeah, see you in the poster session.
Thank you, Clive, for the presentation. And sure, yes, we can have some questions. So the audience, please go ahead.
Okay, this might be a simple question, or we can come back to it. Are you thinking to incorporate other databases like the vasculature I'm thinking of, or maybe more particularly fiber bundles in combination with LGN or MGB?
Yeah, at the end, this is something that I think we have to do. If we want to link it with the function, then the next question is to definitely incorporate the fiber architecture. We can also see some aspects of that when we look at the ventral part of the medial geniculate body right here. It's not clearly visible right here because I've skipped the cytoarchitectonic details, but there's actually a very, very clearly to be seen fiber bundle right here that is also has been part of the delineation itself. So it helped to delineate this ventral part of the medial geniculate body. And also some smaller fiber bundles also help to delineate the different subdivisions from each other. So yeah, to give a short answer, I'm looking forward to do that. Yeah.
Yeah. Thank you.
We have any other questions, Alan?
This is more of a comment in general. I've seen in this session a number of different strategies for improving resolution. And I would question specifically to Kai, how would you think these various strategies will improve the segmentation that you've described here? How can we validate these various strategies for super resolution?
Well, are you referring to higher resolution when it comes to more automatic?
I'm talking about the stuff that you've just seen from Mingli Zhang, the stuff that Christian has done, the various strategies to try and improve on the resolution. And I'd be interested in your perspective on how that might affect the segmentations that you've just showed here now.
Well, using this approach that Christian has developed, we can already see that the segmentations look quite well. So going from 2 micron to 1 micron, I would not expect it to be that much better for such a use case. But when we are talking about cellular segmentations, like Eric has talked about, then I would definitely think that the higher the resolution, the more we can benefit from the information that we can extract from that. But when it comes to mapping the medial geniculate body or lateral geniculate body right now, we've also seen that the the section where it doesn't, the sections where it doesn't work quite well, we think that we should go into 3D. So 3D also means higher resolution again. So a general aspect would be, I would say, yeah, the higher the resolution, the more we can benefit from these very detailed and also sometimes minuscule differences between the subdivisions. Because I've mapped some areas in the BigBrain right now, and this was definitely the most difficult one. And it was of enormous help that I had some automatic procedures that could actually guide and help me to some extent. So yeah, I would say more resolution, definitely better. Higher resolution, definitely better.
Yeah, I could also agree that upscaling in some sense, and maybe going to the 1 micron or even maybe sub-micron resolutions for the BigBrain would like many would benefit from that. And even though we don't have, correct me if I'm wrong, but we don't have any more sections that can be rescanned at lower resolutions. Is that right, Alan?
I think we have, this is, Timo can answer this more directly, but we're at pretty much 75% of the data is being collected at 1 micron. Timo?
Yeah, there are sections available, and there are sections scanned at 1 micron, and we have plans also to release them, but they are not all of the same quality. And then we don't have them all, but a large fraction. And I will actually a little bit come to this in my talk later today. Yeah. But there's 1 micron data available.
Yeah, okay, but I also wanted to point out that maybe if we don't have less than maybe 20 microns or 1 micron available, a model can be trained or on some other data. And even though if we don't have the capacity, the scans to learn from, in a sense, what Mili was presenting, or what Kai was talking about right now, even this knowledge can be transferred, right? So even though the data is not exactly the same, I think it can still produce some sensible results on the upscaling of the BigBrain 1 or 2 or any other data, right? So just a short comment on that.
I would agree, but I also see now where Alan's comment was, or Alan's question was, where was which direction it went. I mapped these structures on the 1 micron sections. It wouldn't have been possible to do this on a lower resolution. So this requires high resolution. And I guess the question was directed into this direction.